This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 330, recorded on March 27th, 2015. Hi, everybody. I'm Vincent Racaniello, and you are listening to TWiV, the podcast all about viruses. This episode of TWiV is sponsored by ASM Press, and they would like you to know about their book, Principles of Microbial Diversity, a brand new textbook all about the microbes that are out there and how they interact. You can find it at asmscience.org. Joining me today here in the TWiV studio, Dickson Despommier.
Hello, Vincent.
Are you all right, Dickson?
I'm better than that, I think. I think I'm in good health, and I enjoyed waking up today and doing my thing, which in this case was going to Fordham and interacting with students in a project that we're involved with in a course that I teach.
It is 7 degrees Celsius here in New York City, and all clouded up.
Yes.
You know it's supposed to snow tomorrow?
Get out of here.
10% chance.
Stop it.
Yeah, it's right here.
We don't need this.
Do you know what percentages are, Dickson?
I do. I know what percentages are.
It probably won't snow. You know, it has snowed on Easter.
Yeah.
Which is sometime in April, right?
It is. It's next weekend.
Next weekend.
Sometimes Easter is way earlier than that, and it can really snow.
No,
sometimes it's in March?
Yeah.
Oh, yeah.
What determines when Easter is?
Good question.
It has to do with the lunar calendar.
Well, since we're skipping introductions.
Yeah, I was going to say, Kathy, you haven't been introduced yet.
It's the following the vernal equinox.
Okay, no problem.
It's okay.
On this episode,
everybody's here.
Everyone's here. Kathy Spindler from southeastern Michigan. Welcome, Kathy.
Thanks.
Rich Condit from north central Florida. Welcome, Rich.
Thanks.
And Alan Dove from western Massachusetts. Welcome.
Good to be here.
I think that's funny. You can talk. It's okay.
I think it's great.
I think it's fantastic.
Something a little different.
So that's a good point,
right.
though, because I do remember when I was young one year, it snowed on Easter, but it could have been in March.
Yeah.
Right.
Interesting. Mm-hmm. All right. Well, we have a lot of material today, so let's. How's the weather elsewhere in the country? Anyone want to file a report?
71F, 21.7 C, high overcast. It's kind of, it's better than where you are. But
25F.
Not a great day.
25F minus 4C.
25. Ooh.
Minus 4C. You're cold.
Mm-hmm. Is it sunny?
Brr.
No, it's not sunny, and we have had a few flakes, but it's not completely clouded over. It's just the sun is trying, but tomorrow it's going to be sunny.
It's funny, Kathy.
I've got 6 Celsius, and dew point is 0, and overcast layer at 2900. So yeah, it's kind of gray.
Yeah. So I took the subway this morning, and I saw a few flakes there too, but that's different.
Last week
2, 3, 4.
I taught a class at Rockefeller, and after the lecture, we had a discussion with the students with papers, and 2 students presented, and they had little PowerPoints. And the first guy, his first slide was the weather report.
Nice.
Isn't that great?
Yep.
It's just wonderful.
Indeed.
All right, a few follow-ups. First of all, Jennifer is doing the timestamps.
Excellent.
So she wrote, shortly after someone suggested to have timestamps, and she said, I would be happy to assist TWiV by marking down the times. I already listened to TWiV on the day it's released, so I could quickly send the information back to you.
Nice.
I'm an avid fan, and I would be happy to volunteer in other capacities if needed. I'm a second-year PhD student of Dr. Palazy's, and I was the one who shared our virus holiday cookies with the TWiV Facebook page last December. So if you will notice the last week's show notes, we have timestamps.
Mm-hmm. Oh, I didn't notice.
Yeah, it's great.
They are there, and thank you very much, Jennifer. And we really appreciate it, right?
Mm-hmm.
Yes,
it's cool.
Absolutely.
It's cool.
Oh, that's cool. Look at that. Cool.
Yeah.
I'll be
Very
there.
cool.
You like that?
Yeah.
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 So the person who requested it, there you go.
Yep.
TWiV, the TWiV Army responds.
Wow.
I'm going to keep that window up because there are a lot of comments on that episode, Dickson.
Yeah, I'll read one of them, too.
Let's see.
I'm wearing my hair shirt today.
Your hair shirt, yeah. You should be.
And the pointy hat. Right?
And the pointy hat, exactly right. I forgot my wand. It's at home, but I'll go back and get it.
Alan, can you take Kevin's email?
Okay, Kevin writes, "Hi, TWiVers. I just wanted to chime in on part of the discussion of TWiV in TWiV 329 when you talked about PKR, which binds double-stranded RNA to phosphorylate and inhibit EIF2 to block protein translation. Dickson inquired about the relationship of this molecule to toll-like receptors, TLRs, and other innate receptors, and Vincent rightly pointed out that PKR is an effector, while TLRs and other pattern recognition receptors are sensors. But this distinction is a bit confusing, since PKR can bind and apparently sense RNA, so why isn't it a sensor?
Yeah.
I had this same conceptual confusion when I began my PhD in an innate immunity lab, and the question is by no means settled. In fact, some people would call PKR a sensor, and cites a paper. Vincent's explanation that PKR is inducible is not quite complete, since many receptors, notably the NOD-like receptors, are barely present at steady state, but are inducible by inflammatory cytokines, and many would consider these proteins the pathogen recognition receptors. My own opinion is that the sine qua non of a pathogen recognition receptor is the ability to induce the transcription of pro-inflammatory cytokines. I can clarify why I think this is a useful distinction if you like, but the fact is no matter how much PKR you activate, you will not get the production of something like TNF-alpha without a legitimate pathogen recognition receptor in the mix. I'd be interested to know if you agree with this distinction or think there's a better way to discriminate. All definitions seem to fail at the edges, but I think the ability to induce inflammation might be a useful conceptual dividing line. A related question in the minds of some listeners might be, why would effectors need to sense RNA if they're only made in the presence of viral infection? The explanation here has to do with an infection in the context of a tissue rather than in a petri dish. Virally infected cells produce interferons, which can activate neighboring cells that aren't yet infected. Those cells can then ramp up the effectors, like PKR, but wouldn't necessarily want to shut down transcription until they sense the offending virus themselves. And Kevin hosts the unpronounceable podcast about immunity.
Autoimmunity.
Autoimmunity.
Yes.
What is it?
Autoimmunity.
No.
Autoimmunity.
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 Immunity.
Wait,
Right.
is it autoimmunity?
No.
Audioimmunity. Because it's a
Audioimmunity.
podcast about immunity.
Audioimmunity.
Yeah.
Listen to the first way
Oh,
I
you
said
don't
it.
want me to separate immunity. Oh, okay, got it.
That's right.
Oh, I'm sorry, pattern recognition receptor. I was saying pathogen recognition receptor.
No one understood.
So
No one understood anyway, is
That's
what you were
what
going
I'm
to say.
saying. Exactly.
You know, our definitions, it's stuff that we make up, and I'm not sure how important it is. I'm okay with PRRs turning on the production of cytokines, and PKR doesn't, so it's not a PRR. It's certainly a sensor, right, because it senses the presence of double-stranded RNA, but it's not in the sense of the toll-like receptors and the helicases, cytoplasmic helicase, so that's okay. I don't mind that.
No.
But I have a lot in the writing of our textbook. I have lots of arguments with our immunologist, Glenn Rawle, about how to name things. So our latest thing is we have intrinsic immunity, and then we have innate immunity and adaptive, right?
Dear.
And the definition for intrinsic immunity, I have problems. Is that it's stuff that's there in every cell. It doesn't have to be. It's stuff that's there in every cell. The induction is a bone of contention. So I don't know. Definitions are really for us to sort of help us, but this is fine. I don't mind this idea that. But Kevin, PKR can be present under other conditions of stress and so forth, because there are activators in the cell, like PACT is a protein that will activate PKR. So it's not only activated by double-stranded RNA, and when PACT activates it, then it induces apoptosis. So under certain conditions, PKR has non-viral functions, right? So whatever. Thank you, Kevin. Kathy, can you take the next one, please?
Sure. Kenny writes, "Hi, everyone. My name is Kenny, and I'm a pre-doctoral fellow studying infectious disease at Rutgers Graduate School of Biomedical Sciences. I just wanted to respond to Kathy's question this week regarding the Bio-Rad Science Ambassador Program. I signed up for the Bio-Rad program back in 2013 while I still lived in Bethesda, Maryland. I now live in Brooklyn, New York, and still have not been contacted by Bio-Rad to teach a class. I wanted to bring to your attention, though, that the New York Academy of Sciences has a fantastic similar program where you can teach an after-school life sciences curriculum to underserved middle school classrooms. We do some great experiments with the kids, including a DNA extraction experiment, but with wheat germ, since New York City public schools won't allow kids to take swabs of their inner cheek. They're worried about potentially sequencing the kid's genome and getting personal information about them. We also do ELISAs, patient zero exercises, and much, much more. Great experience for anyone interested in outreach who lives in New York City. You can find more information about the program here, and he gives a link, so that would be important to include. Thanks for reading my email. I love the show. Kenny. And
Cool.
I was just going to say, there's a program at here at Michigan that some students and postdocs started that has some after-school activities and then 2 capstone Saturdays, one each semester, that my lab and I hope to participate in, but the one that's tomorrow is a conflict for almost all of us. And we were going to do the famous strawberry DNA extraction, which I think is very similar, but I think strawberries are octoploids, so you get a bunch of DNA easily.
Plus, they're a lot more, you can relate to them a lot better in wheat germ.
Yeah.
This is all true.
Yeah.
So what's with Bio-Rad? Is it they just doing?
I don't know.
this-
There must be a Bio-Rad person out there listening.
Come
Get
on,
your act together.
Bio-Rad. Come on.
Well, I'm sure they're limited in what schools they work with.
It has to do with teachers writing in and signing up.
Yeah, right.
Yeah.
So
they may just not have. I mean, they don't list on their website where they have these things.
Yeah, well, you know what? If you're going to do this, you should be proactive. They should go out and seek people.
Right, or they should say we're specifically looking for people in these areas.
Yeah.
You wouldn't have to list the school names, but say we're looking for people in wherever. And if they're not looking for people in Bethesda or Brooklyn, then people in Bethesda and Brooklyn wouldn't bother to sign up and be disappointed.
but if they're willing to drive 75 miles away, they might.
They might, yes.
true.
If they're
It's
looking
true.
for somebody someplace else.
I think it's funny that they're worried about sequencing the kid's DNA. Yeah, it's just going to automatically get sequenced if this is something that would randomly happen. I mean, this is hard to get your genome sequenced. How the hell would this happen?
Well, I did have the question about whether IRBs would be required. And
I suppose,
one of
yeah.
my colleagues said, well, if it's a classroom exercise, then potentially not. But I didn't double check it.
As long
So
as you just
some
collect it.
lawyer in the
Yeah,
school system in New York said, no, you can't possibly take DNA samples. It gets into some liability thing.
Oh, dear. Okay.
There you go.
Strawberries are cool.
Strawberries are cool.
Yeah.
You can even do it with moldy strawberries, I'm told.
Then you get 2 genomes.
Get 2 genomes, right. Dickson, this next one is
Yeah.
long. Are you able to do this?
I'm able and I'm willing.
Because I know you wanted to read this, Dickson.
I do want to read this one because I think
It's directed at you.
It's directed at some comments that I made.
Before you start,
Please.
you know Jenny, don't you, Vincent?
I do. I met her at a CFS meeting years ago, and
Does she have some function at in one of the CFS organizations?
I don't believe so.
Okay.
She just came for one of the meetings and talked
Okay.
about her story. But then she's written a lot to TWiV over the years, and she.
This is an awesome letter. Everybody, listen up.
It's a long letter, but
It's a long letter.
It's worth listening to. This is a great letter.
Okay, here we go. Jenny writes, Dear Vincent and friends, I'm sure we received other emails regarding your discussion on episode 329 about ME/CFS and Dr. Lipkin's latest paper. But as a longtime fan of the show who has lived with ME/CSF for more than 20 years, I could not resist chiming in with my 2 cents. Dickson commented that patients with CFS, or chronic fatigue syndrome, are self-diagnosed, going to doctors who are prone to believing their symptoms. For the vast majority of patients, this is not true. The IOM reports said that 800,000 to 2.5 million Americans may have this disease, but that 80 to 90% of them are undiagnosed.
That's IOM report. That's the Institute of Medicine,
that's right.
functioning arm of the National Academy.
That's right. I have never met an ME/CFS patient who easily found doctors to believe them. Every single one of us has been dismissed or even ridiculed by one or more doctors. We've been told that we are lazy malingerers, that we have psychological problems, or that we just need to reduce stress. It is simply not the case that people feel lousy, as Dickson put it, go to doctor and come home with the CFS label. As Alan noted, there is no doubt that the controversy over case definition has hindered progress in this field. Dickson was right to ask how many diseases are really included in ME/CFS population, and he pointed out that absent or objective test conclusions are preliminary. The IOM made recommendations to address that. First, the SEID criteria are no longer a diagnosis of exclusion. If a patient fits the criteria, then he or she should be diagnosed with the disease. The onus is put on physicians to make differential diagnoses, and physician education will be of critical importance if these recommendations are to succeed. Second, the IOM explicitly stated that the recommendations were based on current evidence and should be revisited in no more than 5 years. They anticipated that new evidence, such as Dr. Lipkin's paper or the studies demonstrating brain inflammation, would change our understanding of the disease's pathogenesis and perhaps validate biomarkers for diagnosis. The Lancet editorial stated that IOM was heavily and negatively lobbied for undertaking this study, but failed to acknowledge that there were legitimate scientific and political reasons for this opposition. First, at the time this study commenced, the IOM had never produced a disease case definition. There was no guarantee how many ME/CFS experts would be appointed to the panel, and the community feared this definition by committee would be created by people who knew nothing about the disease. Second, the ME/CFS field has upwards of 20 proposed definitions, so spending $1 million to create the 21st seemed like a questionable strategy. Third, given the IOM's prestige, we knew that we would likely be stuck with the results for a long time, and if the case definition was flawed, then this would have long-term negative consequences. Fourth, the Department of Health and Human Services pursued this IOM study in secret. No ME/CFS experts were consulted about it, and the department's own CFS advisory committee was not informed of the plan. ME/CFS advocates discovered the contract by accident, and HHS completed the deal over the vociferous objections of advocates and researchers alike. All that being said, it is not true that the entire ME/CFS community continued to negatively lobby the IOM after the study began. Certainly some advocates did so and have continued to object, and not without reason. But many advocates, myself included, gave presentations to the IOM panel and provided written input. In fact, the public access file to the study, which contains all of the written material submitted to the panel, totals over 5,500 pages. In my opinion, there is ample evidence that the panel listened to this public input. The Lancet editorial also mentioned the PACE trial and the Cochrane review on exercise therapy. This brings me back to Dickson's question about how many diseases are lumped together in the CFS category. Depending on the case definition being used, it could be more than one. The Oxford CFS definition in particular is overly broad, as it requires only 6 months of subjective fatigue. A patient population defined by only that subjective symptom is larger and less precise than a more narrow definition like the IOM SEID or the Canadian consensus criteria. In fact, a recent report from an NIH panel and a systematic evidence reviewed by the Agency for Health Care Research and Quality both recommend that the Oxford definition be retired because results from studies of this broad group cannot be accurately applied to the more specific ME/CFS group. This is important because most of the studies in the Cochrane review on exercise and CFS were Oxford studies. The PACE trial also used the Oxford definition to enroll subjects. In fact, most of the studies purported to show that exercise is beneficial for patients with CFS has used the Oxford definition. In contrast, there is a growing body of work, such as this paper by Keller et al., showing that ME/CFS patients, defined more narrowly than Oxford, have measurable abnormal responses to exertion. The case definitions are not interchangeable, and research results should not be extrapolated from one group to the other. Patients with ME/CFS do not just feel lousy. This is a serious disease with serious consequences. I have been housebound and sometimes bedbound for more than 20 years. The most severely ill patients suffer from numerous neurologic symptoms, including ataxia and allodynia. This disease costs our economy billions every year in lost productivity, and yet NIH spends only $5 million a year on research. Even Dr. Lipkin has stated publicly that his grant application was denied after a reviewer claimed biomedical research was unnecessary because ME/CFS is a psychological disorder. The IOM report is an exceptionally well-referenced document and states unequivocally that this is not a psychological disorder. It is my fervent hope that we can leave that controversy behind and move forward with rigorous, well-funded research into carefully characterized patients. This is the only way we will find treatments and get our lives back. Thank you for continuing the conversation about this disease. Vincent, I also send you best wishes for your son's recovery. Cheers, Jenny. And let me just state to begin with that I hope that I didn't mean to say anything that was offensive to anybody out there suffering from this disorder. I only meant to say that we do need a more careful case history definition before we can begin to study it seriously. And I think that Jenny agrees with that definition. But I also sympathize with her over the fact that there's no money for this. And it's too bad. I don't quite understand, though, how the IOM could identify such a broad range of sufferers from 800,000 to a potential 2.5 million. That's a very large spread. And how you would attempt to resolve this opinion, which is an opinion by the IOM rather than a definition, is very difficult to imagine, because I think that just shows that we're all struggling with how to carefully categorize and typify real patients that are suffering from this. That's all.
I've seen figures like that mentioned in literature back when we were talking about XMRV.
Yeah, me too. Yeah,
and if you trace the references back, these are estimates. The reason the range is so huge is that the estimates are derived with usually the most conservative case definition at one end and the least conservative case definition at the other.
Yeah.
So it's, if you stringently define just this particular group and then extrapolate from how many people were found in this particular study in that population, then you would say 800,000. If you take what I assume is probably the Oxford definition or something similar, then you would extrapolate there'd be 2.5 million with this disease, and the numbers could go even higher if you take even looser definitions. So
sure.
The point in bringing out the numbers is to say that this is not, this is not a disease that's affecting a few hundred people. This is a disease that's affecting a large number of people in a big way, and needs to be addressed in a serious way.
Okay, so what is the thing that's needed most to get NIH on point to go ahead and allocate some significant funding for this? What's really needed here?
Well, first of all, significant funding for NIH is one of the things that's
all
needed.
right, let's start with that one as an example.
But I think the part of the explanation for this is that this is a condition where you could say, well, if we're going to pour more money into it, what do we study? We're just now at the point where HHS. HHS is the agency in charge of NIH, and they finally decide, and also CDC, and they finally decided at the top level, this is a mess. We're going to turn it over to an independent government body, the IOM, and have them figure this mess out. And by the way, that is precisely what the IOM is supposed to do. They do these reports where they're trying to collect all the information and come up with a reasonable path forward. So in terms of the funding, I don't know that this is something that's ripe for the kind of effort that was put into, say, HIV, where it became obvious early on that we had the tools to do an enormous amount of science on the causative agent for this disease, and of course, you're gonna put a lot of effort into that. CFS, I'm not saying 5 million is the right number. It seems like a very small sum to put into any substantial condition. But I would not expect at this early stage that it would make sense to put huge amounts into it. How you would get the NIH to put more into it, the answer is going to be they'd cut something else, which is not going to be what anybody wants.
So, Dickson, mainly people objected, and there are some other objections on the TWiV website for this episode, to you saying that these are self-diagnosed people and a few other things. So I think we've made that clear that they're not self-diagnosed at this point.
Mm-hmm. And the ones included in the study that Ian Lipkin did met certain criteria.
Of course.
Okay, so mea culpa and mea culpa.
I just want to comment on Jenny's statement that physician education will be of critical importance. And so with our son, we've had trouble figuring out what kind of chronic illness he has. And one of our friends in town who's a physician, she said, do not have him diagnosed with CFS because no one will then try and help him. Right? She said, don't, because then they'll say, well, that's it, and we can't do anything.
Right.
Which is really unacceptable, right?
And what about what Jenny said, that the people that were on this IOM panel included no ME/CFS experts? Is that, I mean, that was an objection evidently to,
I
by
don't
the
know
community.
if that was an initial objection. They were afraid that would happen. I don't know if that was the actual case.
Okay.
Yeah.
And I don't like the notion that HHS was doing this
secretly.
Why?
Really?
That is not correct.
That is not correct?
No, I don't think that's correct. The HHS doesn't, they can't contract studies secretly. They have to announce what happened, I believe. I haven't looked into this particular case, but often when advocates for something say that a government agency did something in secret, what they actually mean was that this was merely published in the Federal Register and that their particular activist community was not directly notified. The Federal Register is a public document that comes out every single working day, and it'll take you a a very dull quarter hour to just read the table of contents on this thing, which I did for a while. I was, keeping up with it, and it's just reams and reams and reams of stuff. But if you are concerned about what government agencies are doing, and this is something that a lot of journalists actually do keep on top of, and if you're covering policy issues, it makes sense to wade through that and monitor it, because these agencies are not required to call you personally when they're dealing with an issue that may affect you personally.
So it wasn't proactively publicized.
I don't think it was not proactively
publicized
to the group that might care the most, and that was probably a public relations error on HHS's part. Right. I would have to see some evidence that it was done in secret to actually buy that use of that particular rhetoric.
Well, it sounds like Jenny's bottom line is that the ME/CFS community had input in the end.
Yes, and,
one way or
and
another.
Also, I mean, it's HHS. I guess didn't involve their own. They apparently have their own advisory group on CFS and didn't involve them in putting out this contract for IOM to examine the issue. But
that could go either way.
Yes.
You could get criticism that it was an inside job.
Yes.
Right.
and I believe I'm not going to. I can't speak for HHS, obviously, but I think the intent behind this was to get something like an outside opinion while still keeping it within the government.
Right.
All right. Before we move on to our science paper, I just wanted to call attention to two kind of comment opinion pieces that were published this past week. One in Nature called "Don't Edit the Human Germline," and one in Science called "A Prudent Path Forward for Genomic Engineering and Germline Gene Modification." And both of these address the idea that with methods like CRISPR Cas9 and zinc finger nucleases, we will soon be able to modify the DNA of human embryos. And in fact, the Nature paper says it's thought that studies involving the use of these tools to modify human embryo DNA will be published shortly, and they cite a paper in the MIT Technology Review, which is quite an extensive piece on what George Church is doing at Harvard and others. And they basically are saying we should probably think about this carefully. The science paper is authored by Baltimore Paul Berg and many others. George Church is on there, Jennifer Doudna, very prominent people who are saying, we don't know what's going to happen here. We've already had some discussions. We should have more, and they recommend that we discourage germline genome modification, make a forum for discussing this. So I thought we should just mention this. This is important.
So they're saying there should be a pause.
I believe discouraging means I don't think it's a formal pause. It's just discouraging.
No, it's not. It's not a moratorium, but they're saying let's think about what we're doing before we do this.
Yeah, and
they reference the Asilomar conference.
Yeah,
and they have Paul Berg was right on. He was a major player in that, as was David Baltimore. So they're familiar with this.
Yeah. So I don't think it's a bad idea to think about this and talk about it before moving on, because you don't know what's going to happen when you modify an embryo, right?
Right.
And you don't want to make sick individuals from this process who have all kinds of problems or don't live. So I think it's a good idea, and it's kind of the right time because this is really moving forward. So
it is really moving forward. I'm just over the last week or so, especially since that pick that I had last week about the mutagenic chain reaction. And a seminar that I went to yesterday about, the advancements in DNA sequencing. Our ability to manipulate the genome is really stepping into a new age here.
Yeah.
And I think, and it's all happened so fast.
Yes.
And I think that the landscape has the potential to change very dramatically. And it is, there are issues here worth giving some serious thought.
Now, some of you may wonder why I favor this sort of, quote, pause and not the pause on influenza modification research, because they both involve protecting humans, right?
Yeah.
Well, here, the possible consequences are really totally unknown. Whereas I can tell you that if you modify an influenza virus to infect ferrets, it's probably not going to have much of an effect in people. That's my opinion and only mine. And I think that kind of work should go forward. And that pause is really not well considered. But here I think we haven't done these experiments yet, and this is a good time to pause and think about it and maybe come up with regulations or...
Well, there's also, I think, a fundamental difference in what's being done in these two different cases. The experiments that Osterholm et al. Are so exercised about are experiments where people are altering viruses to understand how viruses work.
Mm-hmm.
And not only is the intent different, but the experiments themselves are different, because this is an effort to change something a little bit and analyze the result, and change it a little more and analyze the result. A classic virological approach that's being applied, and that has been applied for decades to these types of problems. Whereas with germline modification, this is not a basic research experiment when we're talking about humans. Because nobody's talking about stopping germline modification of mice. We've been doing that for a long time.
Right.
This is germline modification of humans. That's not an experimental technique that you do for basic insight into how humans work.
It's clinical use, right?
Because you couldn't ethically do anything like that for experimental purposes. Yeah, so this is a clinical application which brings in a whole set of medical ethics, and I think it does away with the concept of informed consent, because you are doing a treatment on a person who cannot possibly give consent, being the one who you're going to make, and you're doing it, even if you say, well, the parents could give consent by proxy, as they do with childhood treatments, they can't give truly informed consent because we don't know what any of this stuff is really going to do.
Yep. Yeah,
I think the other thing that helps in this particular case, too, is that there's at least initially, seems to me a very sharp line that we're drawing saying, don't engineer the human genome, the human germline. Okay?
Yes.
Yeah.
And that really helps to be able to define quite clearly what the issue is.
Heritable changes are the problem.
It's not saying don't stop all this research, period.
Yeah.
Okay.
I think just to point out why people in favor of the pause for so-called gain-of-function research might see this as a parallel is that they have much more of an apocalyptic view of it than many of us do. And so if you want to say that doing this kind of research on viruses is going to change the whole population makeup of the planet, then that may not be so different from the possible apocalyptic view of germline research on humans changing the population makeup of the planet.
Yeah.
The boys from Brazil.
That's a good point, yeah. All right, those are probably open access, I would guess, right?
Strangely, the one published by Nature, which is a privately owned publication, is open access. The one published by the AAAS, which is owned by an association dedicated to the advancement of science, is closed access and behind a paywall.
Come on. Science.
If you want to read the one, I was actually shocked at that, which I think was a very bad mood. I work for Science periodically, and I really didn't expect that they would have this behind the paywall, but they did.
Tell them to open that one up, Alan.
I don't have that kind of pull, but this...
Oh, come on.
Well, we can... we have some listeners, but all our listeners tell Science to open it up.
Yes, because this is an important...
Yeah, this is important.
And Nature opened theirs up, so come on, guys.
All right, we have a paper today about hepatitis C virus published in Science, said journal.
Yes, said journal.
Probably
of the devil?
Journal, fine journal. Yes.
Probably behind a paywall.
I think it is, yes.
Structural basis for RNA replication by the hepatitis C virus polymerase from a group at two companies, Gilead in Foster City, California, and Beryllium in Washington. What a nice name, Beryllium.
Yes.
For a company. Now, this is an article that has what I think are seminal findings, and I'm putting this into my new, the RNA synthesis chapter of the new edition of the textbook. And so I was really interested in this from that standpoint, but it has some broad implications as well. So, of course, hepatitis C virus is a RNA-containing virus with a single-stranded RNA genome of positive polarity. Something like 11 or 12 kilobases long. Correct me if I'm wrong on that. But this RNA gets into a cell. It's plus-stranded, so it's immediately translated to form proteins, among which is the RNA-dependent RNA polymerase. That will then go on to copy the genome from end to end and make minus strands from which more plus strands can be made. And this particular enzyme is very active, and they have a number in this paper. The virus production per day in each infected patient is 1.3 times 10 to the 12th virus particles. That's a lot. So the polymerase is very active. This polymerase, by the way, is called NS5B, non-structural protein number 5B. We'll call it the RNA polymerase. And what they're trying to do here is understand the very early steps in RNA synthesis. We already have a couple of structures at high resolution, X-ray crystallographic derived structures, showing the enzyme just by itself. I believe what they call the apoenzyme, right? Just the protein with nothing in it, no RNA, no substrates, etc. But this doesn't tell you everything about what happens. In particular, they were interested in understanding. So RNA polymerases, or all polymerases, nucleic acid polymerases, look like a right hand. They have a palm domain that has the active site where polymerization occurs. Then they have a thumb domain and a fingers domain.
Is anybody not looking at their right hand at the moment?
I am looking at my right hand at the moment, and I'm hoping listeners are doing the same. And you kind of curl it up as if you were going to palm down, as if you were going to hold something.
Yeah.
And what you're going to hold is the RNA going through your palm between the fingers and the thumb, as that's where the action is, right?
Like a rope. Yes.
I'm not just looking at it, but I'm playing with it. I'm moving it around, flexing it. Yeah, nice.
Right.
Okay.
He's polymerizing.
So all the polymerases look like this, but this one has some extra features that they want to know what they're doing. In particular, there are two sequences. One is called a beta loop. So it's a very short loop of amino acids composed of a beta sheet. And the other is a C-terminal tail. Okay? And the problem with these is that they occlude the active site. And the structures that were solved before, these are right there where the RNA is going to be made, so it cannot be working with these there. So they must be moving at some point. So they wanted to get the structures of some early stages with RNA and substrates, the NTPs in there, that would shed some light on what's going on, right? Now,
my sense is that those structures, correct me if I'm wrong, but don't actually occlude the active site, but shut off if you can, in your curled right hand, define sort of a tunnel that the RNA is going to go through. What those structures do is basically block off one end of that. Is that correct?
Well, I think they certainly block off one end, right? So if you made a product that couldn't get out, but my understanding is in the APO structure, they're really close to the active site.
It's even more occluded than that.
It's more occluded. So there are a couple of stages that this enzyme has to go through.
Okay.
The other thing we need to know is that of all the polymerases out there, they can be broadly classified in two ways: whether they need a primer or not, a primer-dependent enzyme or primer-independent enzymes. So DNA polymerases are all primer-dependent. Pol2 that makes mRNA, that's a primer-independent. Among the RNA polymerases of viruses, the RNA polymerases that copy RNA templates, they can be either primer-dependent or independent. So some examples of primer-dependent polymerases would be the influenza virus polymerase that makes our mRNA that requires a capped oligonucleotide as a primer. Poliovirus polymerase is needs a protein-linked primer. This virus,
I'm sorry. Go ahead.
It's okay. Go ahead.
In this commentary, he makes the statement that when they synthesize, quoting here, when they synthesize a nucleic acid, most RNA and DNA polymerases do not truly start synthesis of the strand complementary to the template strand. Instead, they only elongate an existing RNA or DNA primer molecule. And that strikes me, if we're talking broadly of RNA polymerases, as not correct. Because I think of like Pol1, Pol2, Pol3, coli polymerase, etc., as being able to start without a primer.
You're absolutely right. They start with the first NTP for sure.
Right. So there's a lot of RNA polymerases that can do that.
Yeah.
But in the virus world, it's different, right?
Yeah, there's both cases, primer-dependent and primer-independent. And this particular polymerase of Hep C is primer-independent. It doesn't need a primer. Apparently, you can take a triphosphate and stick it right at the 3' end. So you have to start at the very 3' end of either the plus or the minus strand.
And that has always seemed to me to be sort of one of the conceptual problems.
Right.
Is how do you copy an RNA and get the very last nucleotide?
Right. And that's what they think is the role of this beta loop and the C-terminal tail. That there's some previous evidence that these are involved in positioning that first NTP right at the 3' end of the RNA.
Right.
And that's what they're going to address by getting some new structures. So this paper is all about getting a bunch of new structures, quite a few, by doing some interesting different things. And one of the things they did is they used a strain of Hep C that is associated. It's a Japanese isolate. It's called JFH1. It's associated with fulminant hepatitis, and it makes a ton of RNA. It's really good. So the polymerase must be good. So they used that. Then they used an enzyme from a virus that was selected for resistance to inhibitors. They used a guanosine analog that inhibits RNA synthesis, and they selected a mutant that has 3 mutations that they thought would perhaps allow stabilization of the early states of what's going on with the template and the triphosphates. This has been used, this kind of strategy has been used for some other structures before, and so they showed that this triple mutant can make RNA, and they use that for the structure determination. And finally, they use diphosphates instead of triphosphates, so nucleotide diphosphates, which will go into the active site, but they won't elongate. So they'll make stalled polymerase substrate complexes, right? But
right. So now we've got a hepatitis virus that's really good at RNA synthesis.
Right.
We have a mutant that, because you've selected it for resistance to this guanosine analog, it initiates RNA synthesis more efficiently.
Right.
Right. And now we're going to jam up the initiation of RNA synthesis with these diphosphate.
Right. So we can freeze it so we can look at it.
So we can start more of it and then we can pause it.
And then last thing they use manganese for these reactions. Typically these polymerases need magnesium, and they need that because in the palm of this enzyme, in the active site, there are highly conserved aspartic acid residues that the magnesium ions bind to, and the magnesium in turn binds to the phosphates of the incoming NTPs and help stabilize them there for incorporation. So manganese works also, but you get a lower affinity, and they basically use this to slow down the reaction so they could freeze it. All right, so all those unique approaches we're using, and they use this to derive a number of structures, and the results we'll talk about. So they basically can get crystals of the enzyme, and then they soak the crystals with RNA templates and triphosphates and solve the structures of these. And they found that all these approaches worked. So what do they find? So basically, the first structure they determine, they actually have 5 different structures of a template RNA with the polymerase, with either UDP or CDP or ADP, GDP with a primer in addition. Okay, and they solve the structures of that. And so this is a very early step.
Now this primer is a 1-base primer.
It's a 2-base primer.
2-base primer.
2-base primer. Okay. They've stuck in there triphosphate and an RNA template. Right. And when they look at the structure of this, they see that the beta loop and the C-terminal piece of amino sequence, if you will, they've moved compared to the apo enzyme structures that have been previously determined. So they've moved because we've got nucleic acid in here. Remember, previously those structures didn't have any nucleic acid in them because they couldn't figure out how to freeze the nucleic acid in the substrates. And all these things they've done here freeze them in there. Okay, so they can see when you've got template and substrate in the active site, the the beta loop and the C-terminal linker move. To allow that to go in there. And they're beautiful pictures. You can see they've done a nice job of highlighting this loop and showing how the template and product can get in there when the loop has moved a little bit compared to the apo enzyme. So basically what they think is that this loop, so they have a previous step, which they don't see, of course. They think that the loop helps to position the template and that first triphosphate together, so that this thing can initiate. And then the loop moves a little bit, and then you've got this RNA template product in there. One of the cool things that they mention they say that in these structures, the beta loop is a little disordered. So there are lots of ways that you can tell that, a protein isn't in a perfect configuration in the structure. It's a little bit fuzzy. And they think that's because it's moving even more than it already has. So it's flexible, and they say it's probably going to even move more. So they have structures with the template. They've got 2 nucleotides incorporated into it, and everything looks good. They get all the right interactions are present. So this looks like a biologically relevant structure. They have the manganese ions coordinating the triphosphates. Everything looks great. Okay, so it's a template with 2 triphosphates bound to it, and basically to accommodate that, the loop, the beta loop, has moved a little bit. But they say this is not enough to get high amounts of RNA because this beta loop is still blocking the active site such that a product couldn't get out of it. So we've only put 2 bases on the template. And for it to be elongated, it would have to move through the active site, and the beta loop is blocking that. So they say it must move even more. Okay, so what they do to address this is to take a mutant or an altered protein lacking the beta loop. So it's been removed from it. Now, about 10 years ago, actually, this mutant polymerase or altered polymerase was published. It can't copy RNA unless you put a primer on the RNA already. All right? So taking out the beta loop, if you don't give the RNA a primer, it doesn't work at all. And so that tells us that the loop is important for putting down those first bases, right, on the RNA. So they solved the structure of this beta loop deletion polymerase with the triphosphates and the primer and the template. And they see now that, again, everything, all the structure looks correct, except now there's plenty of room for the product to go out. So they hypothesized that, even though this is a deletion mutant, they hypothesized that the next step after you establish these first 2 bases on the RNA with the beta loop clamping it in, it then has to move out of the way, and then you get this highly processive or high-level RNA synthesis that would then allow you to make lots of viral genomes.
Right. Yeah, so my sense of this is that it really, what I got out of this is that it really addresses, in particular, this idea of how you initiate transcription at the very first nucleotide. Right. Because it's as if the enzyme sort of threads onto the end of the RNA, and the exit channel is capped, right, with this beta loop. Right. So it just threads onto the end of the RNA and can only thread so far, so that first nucleotide is right in the active site. And so now you get started with a dinucleotide, and then you can open up the back end and elongate, and the RNA can move out. Really cool.
They call it a swinging gate, right? Which I kind of like. Now, I think an interesting question is, what opens the gate? Right? So they say the following, which I thought was interesting. As the dinucleotide primer is extended by another 1 to 3 nucleotides, the buildup of tension displaces the beta loop. So now it moves away.
Pushes the thumb out of the way, and now the thing can slide through.
What is this buildup of tension?
Well, if you've got the next nucleotide moving into the active site,
it's pushing,
and you've catalyzed it joining this chain, it's going to push sterically against the thumb.
Yeah.
And that's going to tend to push it out, and there's probably some sort of threshold,
I guess,
that it reaches. And so that this model that they have is that you've got the closed enzyme floating around, and then it picks up the RNA strand, and it picks up a nucleotide, and then the next 2 transitions are thought to be very slow steps,
right,
where it eventually adds another nucleotide, and then it adds another one, and then that pops the gate open, and pow, it just starts
going like crazy,
processively going.
I like it. I think it's very cool.
I don't know quite how to fit this into that question, but I do know that generally speaking, the thinking is that in ternary complexes of RNA polymerase and a template strand and a nascent strand, that the strength of the hybrid in the active site is a really important force in the stability and the configuration of the complex. And adding or subtracting one or two base pairs in that
makes a big difference,
hybrid makes a huge difference.
Yeah, okay.
Okay? So it could be that once you put in the third nucleotide, the whole dynamics of that changes.
Okay.
And I don't know, in an elongation complex, I don't know if they know what the full length of the hybrid actually is in this particular case. In something like Pol2, it's pretty long. It's like 12 nucleotides.
Right.
So it turns out this is probably a universal property of polymerases, RNA-dependent RNA polymerases of viruses anyway, that don't require a primer, that you have a platform. They call it a priming platform, where that first base is aligned with the last base on the RNA, and it then allows the synthesis of a little primer, and then the gate swings open. So apparently in reovirus polymerase, there's a loop that moves out of the way that has a similar function in influenza virus. Now, the influenza mRNA polymerase requires a primer, but there's a slightly different polymerase for genome synthesis that is primer independent, and that apparently also has this kind of a platform for priming as well. Then there's a phage RNA polymerase. So it seems to be a universal mechanism that for these enzymes that don't need a primer to figure out the way that they assemble the triphosphate right at the end of the RNA is to have a little platform that then has to retract, because that platform by definition is going to be in the active site.
Right.
It's pretty cool. I love it.
It also has some parallels with the DNA polymerases that use a protein primer. So I looked up this paper in EMBO Journal from Tom Steitz's lab, where there's a protein primer, terminal protein primer, somewhat analogous to the adenovirus protein primer, that it's positioned in the same place that an RNA primer would be for DNA synthesis, it looks like. And there's a hinging motion that happens as the nucleotides get added that allow this thing to get pushed out a little bit by little bit, but then suddenly it's reached the limit of how much that hinging motion can allow, and it just looks like it kind of blasts out the rest of it, and then the primer dissociates, and the same kind of thing where now elongation can really take place.
Cool.
This also would seem to get around a problem that the virus would have otherwise. There's certainly in the case of Hep C, where you've got a positive sense RNA genome that you want to translate. Mm-hmm. And you also have all this polymerase sitting around that you're now manufacturing that's going to accumulate in the cell, and the polymerase is going to want to replicate that same template. And so if the polymerase is going around, if you've got all these polymerases going around replicating these templates, you're going to cut down on translation, right?
Mm-mm. Yeah, that is an issue.
So this would inhibit, this would kind of restrict the time that the polymerase spends dealing with replicating the RNA, in a sense. I mean, I'm kind of just hypothesizing here, but the idea would be that it's going around with its fist closed, and it can kind of attach to one of these things slowly and pick up a molecule, and then it puts on a nucleotide, it puts on another one, and then, okay, bang, let's replicate, and goes and gets this done, and then it goes back to the closed fist and just hangs around for a while.
So you're thinking that while it's undergoing this slow initiation step, that RNA could be translated.
It could potentially still be translated, right?
Sure, it's possible, because that is a conundrum, how you replicate and translate the same molecules, and people are trying to understand that.
Do you think?
So I'm wondering whether now I'm really talking outside of my expertise here, but I'm wondering whether the beta loop, in addition to helping position the template RNA in the enzyme, doesn't actually contribute significantly to the stability of that whole initiation complex to help it happen. And then that stabilizing effect can be replaced by the stabilizing effect of the hybrid that I mentioned before as it moves aside and the polymerase elongates.
Mm-hmm. Sure. I don't see why not. Mm-hmm. Makes sense. I think that's part of it, that this is stabilizing that initial interaction.
Do any of you think, this is a very naive question.
Yes, I think it's a very naive question.
Thank you. Well, you give me a naive answer then.
Okay.
What I'm interested in is, you're going to study the heck out of this thing and find out how this mechanism works. Do you think they could gain insights into using any new antivirals to interfere with this process then?
Absolutely.
Naively, yes.
Sure,
Naively, thank you.
For sure. In fact, one of the things they do is to use this fancy new way of looking at initiation to study the mechanism of Sofosbuvir, which is one of the new
I remember reading that.
RNA inhibitors that's just been licensed and is being used. And they can see it being incorporated. So that's a chain terminator. It gets incorporated, and it stops synthesis because you can't add new nucleotides. They can see it being incorporated into the growing chain. It's very cool.
And my other question was, if you had this protein isolated, this protein complex isolated, using an atomic force microscope, do you think you could actually watch this happen?
Don't know.
Is that within the realm of possibility here? Because I keep reading about the marvels of being able to look at benzene rings and things of this sort. You can tell carbon molecules.
You must be reading chemical engineering news.
I actually subscribe to it.
I know you do.
I hope that can be done. Because
I don't know, Alan. What do you think? What do you think?
I'd love to see it. Is what I think.
Yeah, I mean, it looks as though it's screaming for a movie.
Yeah.
So, go ahead. Sorry.
From the therapeutic standpoint, this also suggests, now they can look at how an existing drug works, which I think was really just kind of a proof of concept. And I'm sure what these two companies are intending to do is develop new drugs out of this.
Yeah, absolutely.
And now they've identified, you've got these very, very high resolution views of what is obviously a really critically important step.
Here.
If you can make something that will get in there in the APO enzyme and jam it up in that configuration, you could prevent it from even starting. Or, better still, if you could jam it up in the attached RNA configuration, then it would also prevent unaffected versions of itself from attaching, and you could really just kind of lock up replication that way.
Yep.
Rich, in answer to your question about the interaction with the beta loop, they say the structures reveal molecular details of a platform for RNA synthesis in which the tip of the beta loop now buttresses the end of the short RNA duplex.
Mm-hmm.
So there are interactions there, clearly, that stabilize the end of the RNA and the NTP.
Yes. Okay.
So it's very cool.
That's right. I'm thinking back on, I studied these structures. You really have to be patient to look at these things and figure it out. But yes, they talked about specific interactions between residues in the beta loop and the RNA.
Yep, they have pictures of them here with the little dots showing hydrogen bonding or whatever, So I think this is a really nice paper that really adds to our understanding of what's going on here. And it has broad implications as well. So I thought it was cool.
I just wanted to say that if people want to take a look at the science paper, those who have access to it, there's a model figure there. But Vincent's also written a blog post where he's made an even more simplified model figure that I think is helpful to look at.
That'll help conceptualize all this. That's a nice figure.
Sometimes I write a post as a way of preparing for TWiV. It helps.
Yeah, I go talk to my friends about the paper that's coming up. I do the same thing.
I don't have any friends, so
Well, I had the distinct advantage of being in the TWiV studio, so I had Vincent actually go to the blackboard and draw this for me so that I could get a better understanding, and so he did that too. So he's a consummate teacher at every level. I tip my hat to you.
I don't have anybody to talk to but the cat.
How's your Mount Snowplow doing?
You can talk to the cat, but the cat won't answer you.
Yes.
No, I'm sure she understands.
She probably does.
Yeah.
This episode of TWiV is also sponsored by the ASM Education Department. They'd like to turn your attention to a special issue of the Journal of Microbiology and Biology Education, in which there is a 45-article section on the responsible conduct of research and ethics in science. And you can use this to review how ethical you should be, and also to set up a course to teach people about it. It has articles on how you would conduct such training and incorporate ethics into STEM education. and the influences like on obtaining grants and competing and so forth that could compromise research integrity. So you should check this out. Go to jmbe.asm.org and check out this special section. Okay, now we shall do, as usual, some email. And let's start with Alan.
Okay. Denora writes, Dear TWiV team, it's a lovely day here in the Bay Area, 65 Fahrenheit, 18 C. I assume that's the San Francisco Bay Area, not the Chesapeake Bay or Massachusetts Bay Area.
I was going to make the same kind of comment.
Certainly not Hudson's Bay Area.
No.
I'm a bacteriologist, immunologist by training, but started listening to TWiV recently during my 1-hour commute in the craziness of Bay Area traffic. I was inspired by an avid listener from my postdoc era at UT Austin. I've been listening to the podcasts randomly and happened upon 2:30 today. The reason I decided to write is because I now work for the company that produces instruments for microscale thermophoresis that you mentioned in that episode. Small world. It is not uncommon for people to be a little intimidated by this technique, but it's actually quite simple. You can think of it as electrophoresis, but using heat instead to drive the movement of molecules in a temperature gradient. Unbound molecules will move differently from the complex, and that's how dissociation constants can be obtained. Anyway, I've really enjoyed listening to your discussions and opinions on various matters, including alternative careers for PhDs, and also have a much deeper appreciation for viruses. Keep up the good work.
Rich, is that the one where you got into the method and really explained it to us?
I would have to. That particular method, no, I don't think.
No.
The episode may have included, I'll look it up, may have included another method that I got into. That was the, snow in the headlights, I think we.
Yeah,
right. This one was receptor binding by ferret transmissible H5 flu viruses. And they used this.
2:30, okay.
2:30 was Gene Goes to Washington, flew chickens out.
You know, I gave a talk the other day at Penn on communication, and the guy who introduced me, Glenn Rawl, he announced his 3 favorite titles of TWiVs.
Go on.
As part of the introduction. Well, I don't remember them all. I know one was does a gorilla shift in the woods, and I forgot the other 2. But I think that's cool. People have their favorite titles. Yeah. Kathy, can you take the next one?
Before I do, I wanted to say that at the beginning, I wanted to point out a mnemonic talking about show titles and episode numbers, that this is episode 330, and it's going to be released on 329.
Cool.
So,
nice.
Too bad
just sometimes, I don't have dates with them. I only have numbers, and now that kind of marks. I know when 300 was, and now I know when 330 is.
Yeah,
so.
Cool.
Okay, Michael writes, Hello, Vincent and allies. I've just come face-to-face with 2 of TWiV's recurring themes at once. I'm essentially a lab technician at a major research university, although I spend most of my time in the zebrafish facility rather than in the lab itself. And this facility holds fish for 4 different labs that work together. Unfortunately, I've just been laid off because not one of the 4 labs was able to secure their R01 grant, and they've been forced to shed all their paid employees. One technician is moving on to grad school, but none of the labs she's looking at have funding either. This is in spite of the fact that just a few months ago, we had an old grant that we had to spend several $1,000s from on equipment, or else we'd lose it. You'd think that with the hundreds of thousands of dollars we have invested in equipment and animals, the university would have an interest in keeping its labs productive through funding slumps. But along with the recent privatization of the campus parking system, I get the impression that the university is not terribly concerned with the fiscal long term. As long as I've been listening to you complain about it, I've agreed that the structure of American academic research is unsustainable. But now that it has personally affected me, I move to take action. What sort of steps can I even take as a random American schlub? Are there petitions I can sign, advocacy groups I can become a member of, an awareness campaign about the pyramid structure of postgraduate research? I'm almost afraid of what would happen to science funding generally if this became a national political issue, considering the contempt politicians already have for basic research. If solid research having some amusing aspect to it, hello shrimp treadmills, is considered evidence that basic research is overfunded, imagine the damage the budget hawks could do if they latched onto genuine problems as an excuse to axe the whole enterprise. For the second issue, while I'm between jobs, I'm going to have to watch the cost of my medication and might have to switch from an under-patent drug back to a less effective generic. Without naming names, the older drug is a prodrug, while the newer drug is the pharmacologically active metabolite of the prodrug, effective at lower doses and with fewer side effects. Like fantasizing about winning the lottery, I've fantasized about paying less for medication, and I've thought about different incentive structures other than the patent model. My idea is to take the current patent system and stretch it out entirely into the long tail. Instead of a drug's inventor holding a monopoly on its sale for a decade or two, anybody is free to manufacture it. Instead, generic manufacturers must pay a small royalty fee to the drug's inventor, 0.5 to 1% of gross sales. This fee lasts as long as the patent holder wants it to, potentially forever, leaving the drug profitable for the inventor long after the patent would have expired under the old system. I can imagine problems with this system, primarily that it would be biased in favor of large corporations with well-funded legal teams, though what law isn't, and that wealthy corporations could engage in rent-seeking by buying the rights to mature drugs. I imagine rent-seeking could be mitigated by having the royalty rate drop every time the rights change hands, perhaps with some allowance for small inventors selling the rights immediately to a larger company with the resources to enforce the right. Another issue is that by slowing down the rate of return, you discourage the development of incremental improvements, which I know from personal experience can nevertheless be substantial improvements. On the other hand, this works similarly enough to the current system that it should work without drug companies or national governments suddenly becoming more altruistic and setting aside billions to trillions of dollars for the betterment of humanity and with no immediate return. On a lighter note, I have a couple of listener science picks, 2 very accessible computer games about space exploration. First is Space Engine, and he gives the link. It's a seamless scale model of the universe with most of the good bits modeled, from planets and comets to clusters and nebulae to galaxies and black holes. There isn't much to do but zip around and look at gorgeous vistas, but it gives you a visceral feel for the phenomenal scale of the universe in a way I haven't gotten anywhere else. Be warned, you'll need a pretty beefy computer if you want to run it at the highest detail level. It is currently freeware. The second game is Kerbal Space Program, and he gives the link for that. Minimally competent but expressive green aliens build rockets out of junk and try to explore the universe. It's got 2 main parts. The vehicle assembly building, where you slap together a rocket of your own design out of modular parts, and a flight simulator where you fly your rockets and hopefully accomplish missions with them. It's a great hands-on way to learn about rocket science and orbital mechanics without having to do any math. And when you get bored, you can build a hulking monstrosity entirely out of solid rockets and crash it into things. The full version is $30, but there's a free demo that's essentially just an earlier build of the game, and it's not missing any essential features. There's also a classroom edition that makes the physics more explicit, but I don't know much about it. I love your shows, though I've still got a year left to work my way through. So I didn't get a chance to read this ahead of time, and it was a little hard to absorb Michael's idea for the patent structure thing, but I wonder if you guys have comments on that.
Sounds basically like music copyrights.
Is that right?
Yeah, I said it sounds basically like copyrights, like music copyrights.
They go forever, you mean?
Yeah, they go forever, and you pay a small royalty if you want to include somebody singing Happy Birthday in your movie.
Right.
So I think it's not only a viable system; it's one that's used in other businesses. The problem that he refers to as rent seeking, I don't think is exactly rent seeking. I would refer to that as just business. Sure, a company could go around and buy up rights to existing drugs, and that would be fine. And the owners of those rights would be able to determine fairly accurate valuations for them because they would know what the returns would be. So yeah, fine. I would encourage the practice. I think this is actually a really interesting approach.
I don't know how you'd ever change the model, though.
Yeah, this is something that would have to, it would require something we haven't seen in a long time, which would be congressional action.
That's right.
Cooperative congressional action. Yeah.
Cooperative congressional action.
A lot of action, but no.
So Michael, there's your career for you. Head off to Washington.
There you go.
Michael ought to go to law school.
Yeah.
I don't know any. So, with respect to his first issue, what can he do as a random person about the structure of American academic research?
Yeah, well.
Well, the problem is that your university doesn't actually have the money to support everyone who loses their grants, right? They have they have set their budget to take care of what they need, and this is not built into it.
They didn't pay the thousands of dollars on the equipment.
That's right.
That was bought with grants, and the grants were also running the university, because that's how the university's business model works these days. And so you lost your grants, and you're no longer useful to the university business model.
Right. That is absolutely it, and that's what has to change. The universities need to do that, and that's not happening overnight.
Yeah.
No.
So I don't know what you can do. Does anyone know what Michael can do to be an advocate out there?
I got no brilliant ideas.
Just write your congressman and tell him what your problems are and see what happens. But
there must be some groups out there. Maybe our listeners know.
Yeah, I mean, there are certainly plenty of other people complaining about this. It wouldn't hurt to get together with them, if only to commiserate.
Yes,
yeah.
Exactly. All right. Rich Condit, can you take the next one?
Anne writes, Dear TWiV team members, I have been listening somewhat randomly to past TWiV episodes, so I am not quite sure which episode contained a discussion of the politicization of science and its reporting. I was fascinated to see it played out in a recent set of reports regarding the finding of a select agent, Burkholderia pseudomallari, in macaques at Tulane National Primate Research Center. USA Today broke the story and did a nice job of science reporting, in my opinion. And she gives a link to the article. But it was the AAAS coverage that caught my attention, titled Escape of Dangerous Bacterium Leads to Halt of Risky Studies at Tulane. Starting with the title, the report is at best biased. She's referring to the AAAS thing. She liked the USA Today story. Note that the title itself...
Oh, sorry.
What'd you do?
I started the link.
It's okay.
It's okay.
I don't hear it. Note that, let me see, starting with the title, note that the title itself characterizes the studies on select agents themselves as risky, not just the bacterial agent. I found it even more telling that the brief and biased report was concluded with a basically superfluous dig at gain-of-function studies in viruses. And she quotes that, and in October, U.S. officials halted new federal funding for 18 projects that tweak the influenza, SARS, or MERS viruses to make them more pathogenic or likely to spread among mammals. Federal officials and outside experts are now reviewing the risks and benefits of such gain-of-function studies to decide whether they should be allowed to resume. For what it's worth, regardless of what side one is on, they were excellent illustrations of how science reporting can easily reflect and attempt to propagate bias. So I really appreciate that she pointed these this phenomenon out and the contrast between the two articles. This has to do with a bacterium that is classified as a
select agent. Showing up in, as I recall, macaques that are actually being housed in cages that are outside the facility, open to the environment, caged, but it's nevertheless open to the environment. Whereas those bacteria should have been entirely contained in the high containment laboratories, and that created quite a stir, and there's a lot of questions as to how it happened. Still unresolved.
This quote on the gain of function stuff, it's not even right. Tweaking viruses to make them more pathogenic and likely to spread among mammals. It's just, today is Bash AAAS Day, right?
Yes, apparently.
And as Anne points out, that's entirely gratuitous, that paragraph.
Yeah.
Well, I think the the intent of stuff like that is to try and summarize issues that are probably going to be connected anyway in the public's mind. And yes, they will be. This is another lab accident. And these all get classified as lab accidents, whether they're accidents or not. Because that's the rhetoric that's been put out by the folks who are opposed to the so-called gain-of-function studies. Gee, what if it gets out? And then we had these various incidents with CDC labs, and they're reevaluating their biosecurity, and the vial of smallpox found down in Maryland, and this, that, and the other. So those things all kind of get conflated, and I think the intent of that paragraph, not saying that it really accomplished it, but I think the intent was to say, oh yes, we're also aware of this other story, but there could have been a better transition there to, I don't know, to delineate these as very distinct issues.
Yes, and make it a little more neutral.
Because we don't actually know what happened at Tulane.
That's right.
It's worth remembering that Pseudomallii is, I think it's just an ordinary soil bacterium in some places, like in Australia, right?
I don't recall.
I think it's not unusual to find it in soil if you're in particular areas of the world where it's found in soil. Now, this is not one of those areas, so that's the concern, and it did probably get out of a lab, but we don't know how it ended up there.
Myeloidiosis.
Yeah, I'm on the Wikipedia page, but I'm not finding anything about it.
We've done a show on TWiV. I should know this, but I don't remember because I'm 62 years old.
Well,
is that a good excuse, Dickson?
That explains everything from my perspective, at least.
Where's Michael Schmidt when we need him? He could answer the question. Burkhold area.
I'm looking at a PubMed JClin microbio survey where they took
the bacteria causing myeloidiosis are found in contaminated water and soil, spread to humans and animals through direct contact with the contaminated source.
And what's the geographic distribution?
It's especially in Southeast Asia and northern Australia where it's widespread. Tropical climates. Okay. I don't know if it's a natural inhabitant of the soil. It sounds like it's a contaminant, maybe from other animals or people.
But this was not created by people. This thing has a niche in the environment.
Absolutely.
So the question is, how did it end up in the soil at Tulane rather than whatever within its normal niche?
It looks like on Friday the 13th, CDC released the conclusion of their inquiry into this Burkholderia pseudomallii release. I just, in trying to find something else about Burkholderia, I found this. So I'll stick that link here.
So the infection affects animals and human in tropical regions and is caused by the saprophytic environmental bacterium. Dickson, saprophytic.
Feeds on dead things.
Domestic gardens are not only a common source, but they also contain more than other environments.
It's a detritivore.
So, I don't know how it gets there. They say in this that irrigation is linked with the presence of them, contaminated water.
Right. There's a lot of things out there we don't know.
Well, I'm sure it's known, but we don't. All right. Let's see. The next one is Anthony. In TWiV 328, there was some talk of providing the time location of different topics. Why not crowdsource this? Ask listeners to post the time of different topics in the comments. Yes, it's a good idea, and our crowd has sourced it. Thank you. Thank you, Jennifer. Also, it's much easier to find specific spots in the audio by downloading the MP3 and then using a standard player. The in-page player is convenient, but a little iffy when it comes to finding a specific spot. On a separate note, a measles denier in Germany lost in court. German biologist who denies measles virus exists is ordered to pay.
Lord.
Wow.
A German biologist promised to pay 100,000 euros to anyone who could prove that measles was indeed a virus, was ordered by a court on Thursday to hand over the money.
I love this.
I don't think that's a biologist that did that, actually. He might be German, but I doubt if he's a biologist, if he does any reading at all. Sorry about that.
Well,
I would totally deny him the category of biologist.
They're a delusional biologist.
He's a delusional biologist. That's what I'd put that in that category.
He said
that's crazy.
He denied the existence of the virus and said the illness is psychosomatic.
Well, that's nice.
People become ill after traumatic separations. Wow.
Weird.
So does anyone know the outcome? Did he pay up? Probably not, right?
I don't know. I don't know anything more than that.
All right.
Well, he apparently,
Weird.
The story seems to indicate he's sticking to his guns.
But of course
he would. He's not going to believe anything. Right?
No, he's going to appeal this probably.
You bet.
And he doesn't want to pay 100,000 euros.
He doesn't want to pay 100,000. He shouldn't have offered the bet then.
Well,
too bad somebody didn't figure it out a while back. It would be worth more in dollars.
Yes,
he wants attention.
Yeah, but you better hurry because it won't be worth more than dollars.
Dickson, can you take Varun's?
I will give it a shot.
Try not. Do.
Okay, here we go. Varun CN writes, "Greetings, professors. Recently, I happened to have read an article on Ebola transmission in MBio. The article several times brings down the plausibility of aerosol transmission. I find from previous TWiV discussions that there isn't compelling scientific evidence for assuming that there has been an undetected Ebola aerosol transmission. This seems to be a very troubling point. Being one of the most read articles, it worries me of what the paper finally is trying to impress. The issue has been discussed at great length in TWiV Ebola series. However, I would like to know TWiV host opinions on how far we could weigh the arguments made in this paper, or is it that I have missed some crucial point in the paper?" And then he lists the paper. "Thank you for clarification." Anybody want to comment on that first?
I think we already talked about this one.
Yeah, ad nauseum. That's true. Okay, thank you for the
this particular paper, though, it's Osterholm's hype on be afraid of aerosol Ebola, and it's going to come and get us.
But at least you don't have to be afraid of measles. Tell him to check with that German biologist.
Yeah, this is from back.
I mean, if he's got to believe us, he's got to believe all of us.
This is from back in mid-February.
The paper is titled "Transmission of Ebola Virus is What We Know and What We Do Not Know," and basically all of the aerosol transmission studies or ideas come under the "What We Do Not Know" heading, and it's all what if stuff, basically.
Yeah.
I actually didn't bring this onto TWiV because we had talked about it previously, right?
Yeah.
And then I just thought that this was rehashing everything.
Yeah.
So what Osterholm did is he got 20 people to co-author the paper to give it some kind of cred. And I wrote a blog post, which you can look at, and Alan is aware of, which I said basically this is nonsense. And I emailed the editor of MBio and complained to him, and he said, "Vinny, where else are people going to speculate but here in MBio?" Come on.
All right. There's more to the letter, though. It says, "I also have a science pick to share. I'm not sure if this has been picked up before. It's a neuroscience book called Incognito by David Eagleman. It is in the simplest sentence, the book is awesome. Thank you for ever continuing public education. Twix fan."
I haven't heard of it. Has anyone?
No.
I'll try it. Nope. Varun.
It's awesome.
Varun's from Bangalore. All right. That will do it for email. We don't have that many left, actually. We'll hit those next time.
And by the way, I looked at the CDC report that Kathy referred to while we were reading the other emails, and it looks like they admit up front that they don't have a definitive answer, but they did find lapses in the biosecurity at the facility that is working on Brickled area there at Tulane, and that there are plausible mechanisms by which it could have ended up in the primate facility.
All right.
So it does seem to be a lab release.
Our final sponsor for this episode of TWiV is the meetings department of ASM. In 2016, they are combining the general meeting and ICAC under one roof. It's going to be called ASM Microbe, the largest gathering of microbiologists from across the globe and the premier global event showcasing the best microbial sciences in the world. The only meeting in the field exploring the complete spectrum of microbiology with a true transdisciplinary experience. It offers all of the high-quality programming of both the general meeting and ICAC, targeted transdisciplinary sessions, a profession of microbiology track, a poster hall, topic-based networking, track-specific lounges. That's where Dickson would be. Opportunities for continuing education and more. Nearly 200 sessions and over 5,000 posters against 7 program tracks. A unique forum for interaction among the global community of multidisciplinary scientists. There is a call for sessions. It's going to open on April 7th and will last till May 19th. You can suggest things that they should be doing at this meeting. And to do that, you can go to asm.org/microbe2016. You can submit your ideas for the sessions, so check that out. It'll be a big meeting, and that's going to be in Boston in 2016. Hey, maybe we'll do a TWiV there, Alan.
Yeah. That would be fun.
I just have one comment. I wish they could have thought of a different name besides Microbe, because that's the name of the weekly or monthly journal magazine that they put out now.
Yeah, that's true.
So
I don't particularly like it, but that was a committee developed name. They got suggestions from people, and then some people got together, and that's what that they came up with. I don't know what you'd call it.
Yeah,
but that's fine.
Is there any chance that the BSL-4 facility might be open by then?
Oh yeah, I think so, Dickson. Absolutely.
Hope so.
That's over a year from now.
Yeah.
Should be open. They're going to have a little ceremony when it opens, and we might do a TWiV there.
That'd be fun.
Well,
yeah.
I was absent from the last one. I'd like to go.
No, you can't. I'm not letting you because
I
you
want
always
to put
cancel.
on a spacesuit.
You always cancel.
I want to put on a spacesuit.
Well, and Paul said that you're the only one of the TWiV people that he hasn't met yet. Paul Dupree. So that would be a reason.
I would love to go up and meet him. Yeah.
I just saw him last weekend. He and I were both giving talks out at Penn State, and he gave me a deck of pathogen cards.
Yeah, I got the same deck of cards.
You got the cards.
And
They're all in German.
I sent mine I did my best at translating the instructions, and some of the instructions still don't make sense. So I'm waiting till I get reply back.
I looked up Quartet, which is apparently a popular European card game, and I think I have a pretty good idea of how this thing is supposed to be played now.
When you figure it out, let me know.
If there's 2 or more players, there's one way, and if there's 3 or more players, there's another way. But a couple of the aspects of the game rules are just not quite there. So when I get it completely translated and somebody confirms for me how they think it works, I'll let you know.
It looked to me like the deck could be used for an ordinary game of Quartet if I understood the rules of Quartet correctly.
Mm-hmm.
But I don't know if that was the intent.
Could we play this on Skype, or we have to be together?
I think we would have to be together because you have to see the cards that are put down, and it has to be with one deck.
All right. So next time we're all together, we'll play. That'll be episode 1000 probably.
We'll play Quartet. Yes.
I got a game from a company in the Netherlands, a science game. They sent it to me to try, and I want us to all try it out, but I don't know when we're going to be together, so maybe I'll just send it to everyone. You can try it on your own.
Yeah.
All right, let's do some picks.
Okay.
Alan, what do you have for us?
I have an audio recording, which if you're not accustomed to listening to HF radio, is probably going to be really spooky to you, but that could be fun too. It comes with a little article explaining what it is. So, as you know, I'm into ham radio, and one of the things that's interesting about talking on the radio, especially on the lower frequencies, is you can talk all around the world, and a lot of the propagation of the signal depends on what's going on the sun. So sunspots and flares and corona all have different effects on the radio transmissions. And so what this is some... it's a recording. Somebody happened to be doing an experiment where he was looking at propagation. He was recording something digitally on the air, and simultaneously he was recording a conversation that was going on between a couple of hams. And it's typical ham conversation. They're just kind of yammering about stuff. But what happens is if you do this through headphones, in one ear you've got the time signal from WWV, the National Institute of Standards time signal telling you what time it is. That's kind of dim. And then in the other earphone, you've got this conversation going on with the hams. And then at a certain point, you just notice the static level behind both transmissions just starts to rise and rise and rise. And finally, toward the end of the recording, everybody fades away into the noise. And what happened was we had a class 2.1 X flare on the sun on March 11th. And this is an actual recording of the flare happening and the exact time that it happens, and you can hear the dramatic effect that it has on HF propagation. And this type of thing can also affect things that are more serious than ham radio, like GPS and potentially your cell phone and the rest of the modern world.
Cool.
I thought that was cool.
It's neat.
Cool.
Very neat. Extremely. Kathy, what do you have for us?
I picked an article about the fact that Nature is now going to have double-blind peer review for a few more of their journals. In June of 2013, they started doing it with Nature Geoscience and Nature Climate Change, which are not journals I've ever picked up, but just pointing that out. But now they're Nature Immunology and Nature and other monthly Nature journals will do this, where both the authors and reviewers are unknown to each other. And the responsibility to render the manuscript anonymous falls to the authors, and in some situations that's not going to be possible. So authors can just choose if they want to have this. They say that for these two journals that have been doing it, the uptake has been lower than the enthusiasm that was expressed in surveys. No more than a fifth of the monthly submissions are going the double-blind route. But it's just something that Nature is trying again, evidently, because when I read this the first time, I remember they said that they tried it once before. Open peer review, they tried in 2006. Oh, open peer review. So that was
peer review.
Kind of the opposite. So anyway, this
the
double-blind
problem they had with open peer review was nobody was willing to say anything.
Right. It's all too...
Excellent.
It was, yes, this is an interesting paper. Uh-huh.
It seems to me that making a paper anonymous would be very difficult.
Yes.
Yeah, it depends on how high profile it is, I get lots of papers to review, and if I didn't see the authors, I would have no idea who's doing it. But those are for smaller journals, but Nature is going to get high profile. Why in some cases, Kathy, was it not possible to do it, as you mentioned?
Oh, well, just for the reasons that you could imagine, that keeping their identity secret is impossible if it's a specialist community.
Okay.
Yeah, and if you're following on a previous study from your own lab, and you reference that, but I suppose you would say, you wouldn't say it was your own lab,
Yeah.
And if you use the numbering system for references, unless the reviewers go check every single number, if they just read it and the text itself, it could work.
I also, maybe someone could pull the names out of the PDF also. It must be embedded in there, in some way. Someone could rip it open and find out. It depends on how they...
Yeah. Hopefully they would have a robust system for keeping the anonymity.
Hopefully. One can only hope, right?
There is no unhackable system, but...
Right.
It's true.
Hey, Dickson, what do you have?
I'm watching it right now as we speak. I'm sorry that I was diverted from my attentions here, but I was fascinated with this website. It's a drone camera that's flying inside of the world's largest cave in Vietnam, and
this is really cool.
The most amazing set of photographs I've ever seen in my life. I've been to Vietnam. I didn't even know this cave was around. If I had known that, I would have gone there. Trust me. I didn't even know the world's largest cave was in Vietnam until I ran across it. But it's just an amazing thing to watch. It's got beautiful background music, and the video is just riveting. So I just thought it was entertaining and soothing, as well as amazing and perhaps awesome.
At that scene where they're coming into the big chamber and you see, there's a rock with some algae growing. Wait, those are people standing.
That's right. This is a very, very, very large thing.
Yes.
Yeah, it's amazing.
It's beautiful, too. It's amazing that this remained untouched by the conflict that they had.
It's probably in a remote location.
Well, yeah.
Would be my guess.
Still.
Thank you, Dickson.
Oh, you're welcome.
Rich Condit,
Cool.
what do you have?
I've been looking for an excuse to call the Volvo Ocean Race science ever since it started. And it does have quite a bit of technology in it, but but now I can finally justify this because they've dropped some research buoys in the Southern Ocean. But let me just say that the Volvo Ocean Race is another one of these round-the-world races, and the composition of this one is it's a one design race, so all the boats are identical. They're 65 feet long. They're of a special class of boats that are kind of flat bottom, twin dagger boards, twin rudders, and a tilting keel that are designed to, surf downwind on big waves in the Southern Ocean. They're just incredibly high-tech machines, and all the tracking and communication is very high-tech as well. In this particular race, it's done in several legs where they get to stop in between and sort of lick their wounds. And it's also the crews are there were 7 boats started, but one of them crashed, and 6 of them, the 6 remaining. 5 of them are all male crews, and there's 9 males on the boats, and one of them is an all-female crew, and there's a dozen women on the boat. And they're sailing around the world in this race, and they're right now in the Southern Ocean. A couple of days ago, they were on the planet at a point that is as far from land as anywhere you can get on the planet. And as they pointed out, they were probably closer to the International Space Station than any point of land. And since nobody ever goes to the Southern Ocean, they took the opportunity, each of them carrying a research buoy, to drop into the Southern Ocean and track all kinds of stuff. And so this particular video shows what it's like in the Southern Ocean, which is just a horrendous place. And you get an idea.
That tells you why nobody ever goes to the Southern Ocean.
You get an idea. I mean, we're talking 50-foot waves here and 30-knot winds. I can't believe they sail in this stuff. They're getting knocked down and all kinds of awful stuff.
And by the way, the crews for these boats are regardless of what nation they're ostensibly representing, they're overwhelmingly Australians and Kiwis.
Mm-hmm. And so,
just while we're talking about the crazy people doing this,
the one other thing that I would add that I really like is that they interview the people. They talk to the people on the boats as they show them throwing over these buoys, and they ask one of the guys, why are we launching this buoy overboard? And the guy says, it's to make the boat lighter.
That's good.
So at any rate, there's a great website where you can track this race, and I'm addicted. It's really good. And now's the time. They're going to be rounding the horn in the next few days.
What are these buoys going to measure? Do you know?
All kinds of stuff. I don't know. Drift, direction, ocean currents, compositions, temperatures.
They did a paper on TWiV a while ago where these guys they drop buoys in the oceans and they filter out the microbes, and then right on the buoy they extract DNA and start PCR reactions.
No kidding.
It's just so cool. They're specially made for these guys to do this, so they get it. And these things are floating around freely. Right, they're not anchored buoys. They're floating around, and they go with the current. There's this big, what is it, the gyre, the North Pacific gyre, I think.
Lots of gyres out there.
And they go in circles, and they know where they are. They have radios, but they let them go around this gyre a few times, and then they pick them up. That's so cool.
Yeah.
The stuff you can do. But Rich, I bet you'd like to go on one of these boats.
No. I'd like to go on one of the boats. Yes, on a nice day.
Not on a race.
There ain't no way I'm going near the Southern Ocean or some of the other stuff they've been in.
Don't they call it the Roaring 40s?
Absolutely terrifying.
Isn't that the Roaring 40s?
Yes.
Okay.
Yeah.
I thought you would. You want to go to Mars, man. You don't want to do this?
No, this is just absolutely. I've been too close to. Well, not even close to something like. I mean, these guys in the middle of the night are doing accidental jibes and getting knocked down. Okay, so the boat is like at a 90-degree angle, and they're scrambling to get everything back together to get the boat back upright. In, like I said, 30-knot winds, 30-foot seas. I mean, forget it. It would just be absolutely terrifying.
Well, the boats are also constantly on the brink of falling apart.
Yeah.
Because they're built to be as light and as fast as possible within the constraints of the one design rule. And so everything's breaking all the time, and they're constantly fixing it, and it's really crazy.
All right. So Rich is not going on these boats. Okay.
Good. We'll still be a TWiV team then.
My pick is an announcement by 3 philanthropies: the Howard Hughes Medical Institute, the Bill and Melinda Gates Foundation, and the Simons Foundation, who are launching a new program to give support to promising early career scientists. These are 3 foundations established by billionaires, and they are going to put $148 million over the first 5 years to give 70 grants every 2.5 years to scientists with potential. So there is a website we will link to. You have to be on the list of eligible institutions, and they give you information about how to apply. So I think this is good. I think they need to up it about tenfold. And then
Add a zero, right?
Then they can make an impact, because someone has to replace what NIH is not doing, especially for early career people.
Well, there's another, the Science Philanthropy Alliance, which is some of these same organizations, is now getting together to try to draw more philanthropic funding into science. I just did a story on science funding for Nature Medicine, and this is consistent with that effort that these foundations are also now doing this direct. And by the way, I didn't know Marianne Carlson was at the Simons Foundation now. I just saw that in this press release.
Yes, she's been there for quite some time.
Old news, I'm sure.
She has. So she took a sabbatical a long time ago and started working there. It's just downtown in New York here.
Right.
And then she still has a lab here.
Okay.
And sort of did both, which was okay with the Simons Foundation. And now it is my understanding she will be there full time. She's closing her lab down.
Okay.
And is going to be doing that. And in fact, so she has tried to broaden their interests into microbiology. And I gave her some links, and they ended up supporting this program I told you about, where they drop buoys into the Pacific and measure the bacteria. That's one of the programs that they're supporting in her portfolio, so it's pretty cool.
Cool.
They just hired Boyana Conforti, who used to be the editor-in-chief of Cell Reports, the open access journal, okay? And she is their director of science, communication, and outreach.
Cool.
So we'll see what she does, but maybe they'll support podcasting. We'll see.
Yeah.
We had a couple of listener picks from Michael, which you heard previously, and that will do it for TWiV. Excuse me, TWiV 330. What a nice number, 330. And this will be at iTunes at twiv.tv, and always free. All the back numbers are there. Check them out, 330. And if you have questions and comments, send them to Dickson at TWiV.
We'll be happy to read them.
TWiV at twiv.tv.
That's right. Bring in my hair shirt next time.
Now, what is this hair shirt?
Well, that means that you're suffering from something that you thought you might have done, a guilt thing, and this takes it away.
It's a form of penance.
A form of penance. Yes. So I might have misspoke.
Vinny, I thought you were Catholic.
I might have misspoke the last time we had a podcast, so I tried to correct that.
It's okay, Dickson. You tend to talk wildly.
No, I just speak my mind.
You don't filter, do you,
Dickson? I speak
Dickson?
my mind. I certainly do. Are you kidding?
You do filter? Yeah.
No, knowing what was on my mind.
You'll be glad he does.
You'll be very glad.
All right.
I do.
Thank you, Dickson.
You're welcome.
Evidently, the Catholic Encyclopedia calls it, or it says it's a garment of rough cloth made from goat's hair and worn in the form of a shirt, or as a girdle around the loins, by way of mortification and penance. And then there's some pretty ugly images, too.
Yeah, well, okay, so I take it all back then.
Hey, Alan. Jeez,
Yes.
I called you Aiden. Alan, I was raised a Catholic. However, I have no longer any connection. As soon as I left home, I stopped.
You gave it up.
I gave it up, for sure. And my kids are not raised anything. Dickson Despommier can be found at verticalfarm.com and urbanag.ws. Thank you, Dickson.
You're welcome.
And Rich Condit can be found at the University of Florida in Gainesville. Thank you, Rich.
Sure thing.
You can actually go right to Rich's website by typing bit.ly/poxdoc.
Yeah, you did that for me.
A long time ago.
Long time ago.
When you retire, you're going to get rid of your website?
I think it will be trimmed down to something that has my mugshot and says I'm emeritus professor.
Oh, that's too bad.
unless I craft something else myself. I'll tell you, I don't know what's going to happen when I retire, and that's the magic of the whole
Come
thing.
on in with me on the vertical farm. Come on, we can do this.
I know one thing that's going to happen.
What's that?
Is that you're going to continue TWiVing.
Yes,
Exactly.
indeed. As long as you'll have me, I'm here.
Always.
Dude.
Always.
Forever.
Yes. Kathy
There's
Spindler.
going to be a lot of TWiV from the road, I'll tell you that. That's okay.
We'll have to get you a mobile setup.
Charles
That's
Carroll
okay,
of
I can do it.
TWiV.
No problem. Kathy Spindler is at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is fun. And I was trying to figure out how to do the opposite of exiting before my time from the show because I entered before you introduced me today. So I haven't figured out how to do it yet.
That would be cool. That would be existential. Before we connected, Dickson was looking at the show notes, which she never does.
Stop it.
And he said, what is this southeastern Michigan? And I said, it's to tell me where Kathy is because I forget. And he said, it's not in southeastern Michigan. So I brought up a map and I showed him.
Okay. So it's.
And then I said, should I move to Michigan? Because I'm looking for a place to live. I don't want to die
Ann
in
Arbor
New
is a
Jersey.
beautiful place.
He said, but I said, Kathy and Michelle are there. I can't go there. That's not good.
Oh, you can go to Michigan. There's lots of great places.
So I said, what about East Lansing? And he said, no.
No, definitely not.
Okemos.
Correct.
Yeah, that's what he said. All right. Anyway, Alan Dove can be found at turbidplaque.com and also on Twitter. Thank you, Alan.
Always a pleasure.
I'm Vincent Racaniello. You can find me at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.